Hello, my name is Ghulam Sadat, and in this brief video, I will be talking about muscle contraction. Muscle contraction is a complex concept, but in this video, we will try to simplify this concept by explaining what is neuromuscular junction, what components are involved in a muscle contraction, and finally, if we could summarize and identify the major events of skeletal muscle contraction. We need to know that a skeletal muscle fiber contracts only when stimulated by a motor neuron. Hence, each skeletal muscle fiber in our body is functionally connected to the axon of a motor neuron. This functional connection is called neuromuscular junction or synapse. The three components of a synapse are motor neuron axon, cell membrane of muscle fiber, which is also called motor end plate, and a small space between these two, which is called synaptic cleft. Neurons communicate with muscle by releasing chemicals called neurotransmitters. Muscle cell, which is also called muscle fiber, is just like any other cell. It has a cell membrane, it has a nucleus, and it has cytoplasm and organelles. However, a unique feature of muscle cell, and especially here we're talking about skeletal muscle, is that it has contractile fibers called myofibrils. There are thousands of myofibrils in a muscle fiber, but they are arranged in small units, and each unit is called sarcomere. The bottom right picture shows the two contractile myofibrils, which are also called thick filament and thin filament. Thick filament is made of myosin molecule, and thin filament is made of actin, troponin, and tropomyosin. Muscle contraction occur actually due to interaction of thick and thin filament. This results in shortening of muscle fiber, which then pulls on the body part that it moves. A muscle fiber does not contract until a neurotransmitter is released. When an impulse reaches at the end of the motor neuron, the neuron releases a neurotransmitter called acetylcholine in the synaptic cleft. Acetylcholine diffuses across the synaptic cleft and binds to receptor on muscle fiber cell membrane. This causes generation of electrical impulse, also called action potential, in the muscle fiber. The action potential travels deep in the muscle fiber through transverse tubule and causes release of calcium from sarcoplasmic reticulum. Then calcium further goes ahead and binds to thick and thin filament, and that causes sliding of these two filaments over each other. This is quite hard to understand, but in order to visualize this process, let's watch this animation.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm.
Okay, so once the calcium is released from sarcoplasmic reticulum, it binds to troponin. This leads to displacement of tropomyosin and exposure of binding sites on actin. Subsequently, myosin binds to actin and muscle contraction occur. Let's watch another animation to understand the interaction between thick and thin filaments.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
After muscle contraction occur, there are certain steps needed in order for the muscle to relax. First of all, there is an enzyme called acetylcholine esterase in the synaptic cleft which decomposes acetylcholine. Remember that acetylcholine is the neurotransmitter that initiates action potential in the muscle fiber. So we need to get rid of that. Second, calcium is also reabsorbed back into the sarcoplasmic reticulum, because as long as calcium is in the cytoplasm, it will cause cross linkages between actin and myosin. And finally, ATP is needed to break down cross-bridge linkages between actin and myosin. As you can realize here that ATP is not only needed for muscle contraction, but it is also needed for muscle relaxation. After these steps, actin and myosin is relaxed and they are ready for the next round of muscle contraction cycle. This concludes our session on muscle contraction, and I hope you learned something.